Hello students, welcome back to biology classes. Today we will discuss on very simple but very important topic, that is sex determination, from the chapter 5. In the cells of every diploid organisms, there are two types of chromosomes, autosomal chromosomes and allosomal chromosomes. Autosomal chromosomes are always in pair form, whereas allosomal chromosomes or sex chromosome, they can be either in pair form or there can be a single sex chromosome in the cells of an individual. And if these sex chromosomes are in pair form, they can be either similar or dissimilar. Okay? So sex determines the hereditary differences between the two individuals of the same species. That means we can find out which one is male individual and that of female individual. And it is determined at the time of fertilization, wherein the male and female gamete fuses together and we come to know whether an individual is male or female, right? So we can say, based on the sex chromosome, we can identify whether an individual is male or female. The clue on sex determination was based on the cytological observation that was done by Henking in 1891. And this study developed the concept of genetic or chromosomal basis of sex determination. That means this was the time where the scientists came to know that chromosomes are the one which can determine the sex of an individual. Henking in 1891, he found some nuclear structures when he was studying on spermatogenesis in some insects. What is spermatogenesis? It is nothing but the formation of male gametes, and those are the sperms. So we can call the spermatogenesis as gametogenesis too, since it is the formation of the gametes. Henking found some unique structures when he observed under microscope inside the nucleus. So those were the chromosomes which were there, but he found some unique pattern or structures inside these nucleus. He observed that 50% of the sperms received one structure, okay, whereas other 50% of sperms did not receive that structure, and that structure was nothing but some X-shaped bodies which he found in one sperm, that is 50% of sperms, whereas in the other sperms he did not find this X-shaped structures. So he named these X-shaped structures as X body. But later on it was identified as X chromosome, which is nothing but the sex chromosome. In 1902, Stephen identified Y bodies in the nucleus of some species. So Y bodies were the small structures that were seen inside the nucleus. In 1905, Wilson and Stevens, okay, these were the two scientists which they put forward the chromosomal theory of sex and named these X and Y bodies as X and Y chromosomes, which are the sex chromosomes or allosomal determination of sex. Okay. So the chromosomal basis of sex determination was based on the formation of such different kinds of gametes in an individual, okay, and that was present in one of the two sexes. So since there are two different types of gametes that are formed in an individual of same species, and that too one of the two sexes, it is also called as heterogamesis. Hetero means different kind of gametes. So there are the male heterogamesis as well as the female heterogamesis. So today we will be learning the heterogamety or heterogamesis in males as well as in females. In male heterogamety, there are two types of gametes that are formed. Okay, in one gamete, one type of gamete, there will be autosomes and one X chromosome. Okay. Whereas in other gamete, there can be autosomes and without this X chromosome, or there can be autosomes with Y chromosome. Okay. So there are two different types of gametes that can be formed in an individual. So based on male heterogamety, there are two types of heterogamety which we will be studying today: XO type and XY type of sex determination. In such species, the females will have similar kinds of sex chromosome, that is XX. This we should remember, whereas males will have different kinds of sex chromosome. Okay. So the first one of male heterogamety is XX and XY type of sex determination. Okay, and this we see in most of the mammals including men, as well as in most of the insect, for example Drosophila. And here the males have one sex chromosome which is smaller in size from that of other sex chromosome. So the larger one is the X chromosome, okay, whereas the smaller one is the Y chromosome. Okay, so X is larger than Y chromosome, and since we see there are two gametes that will be formed here, two types of gamete, one which will be having X chromosome and other gamete will be having Y chromosome. Okay, so since there are different kinds of gametes that are formed, or different types of sex chromosomes are present in this individual, that's why we call it as male heterogamety, wherein the males will produce two different types of gametes and females will produce same type of gamete or eggs, since these individuals have similar kinds of chromosomes. Okay. So therefore we call females as homogamety. The genetic analysis, when it was carried out, it was proved that this Y chromosome is meant for to determine the maleness in human and other mammals. That means we should remember that Y chromosome is only meant for maleness, but it is not in the case of Drosophila. So we see here that X chromosome is larger than that of Y chromosome. With respect to sex determination in human, we see that males will have X and Y chromosome in their cells along with the autosomes. So there will be the gamete which will have X chromosome, and those are called as sperms, okay, X sperms, and Y, the gametes which have Y chromosome are called as Y sperms. So the males will produce X sperms and Y sperms along with the autosomes that will be present in these cells, whereas the females have similar kinds of chromosome, sex chromosome, that is XX, will produce same type of gamete with X chromosome along with the autosomes. So these are the eggs. Okay. So since males produce two different kinds of gametes, we can say that 50% of sperms will carry X chromosome, right? And other 50% of sperms will carry Y chromosome. And the sex of an offspring or individual is determined at the time of fertilization. So we have 44 autosomal chromosome, okay, with one pair of sex chromosome. So males will have 44 chromosome with X and Y sex chromosome, and females will have again 44 chromosome, autosomal chromosome, along with one pair of sex chromosome with XX chromosome. Whereas in males we see there are two kinds of sex chromosome, X and Y. So when the sperms with X chromosome fuses with the egg will produce females, and the sperms having Y chromosome, when it fuses with the egg, will produce males, right? So we know that 50% of sperms will carry X chromosome and 50% of sperms will carry Y chromosome. So therefore there are 50% of chances or probability to have male or female offsprings to that parent in a particular mating. Next is XX and XO type of sex determination. So here we see female homogamety with similar kinds of sex chromosome XX, whereas male heterogamety we see here there is only one sex chromosome which we see in an individual. O denotes that there is the lack of one more sex chromosome. So therefore we call this type of sex determination as XO type of sex determination. Okay. So the examples here are grasshoppers, cockroach and roundworms, wherein we see such type of sex determination in the individuals. So since there is the absence of one sex chromosome, definitely the total number of chromosomes will be less in males than that of females, right? Since one chromosome is absent here, sex chromosome. Okay. So we'll take an example of grasshopper. The autosomes, total number of autosomes are 22 in both male as well as females, whereas in male there is one only X chromosome, sex chromosome, and in female there are two X chromosome that we see here. Okay. So here 50% of the gametes which are released by males will have the autosomes along with one X chromosome, whereas other 50% won't have or lack the sex chromosome. There will be only autosomes which will be present in this gamete. So the sperms having X chromosome when fuses with the egg will produce females, right? Whereas the sperms without sex chromosome, there are only autosomes that are present. When they fuse with the eggs, what will happen? There will be males that will be formed here. Okay. The second type of heterogamety which is seen in females. So therefore it is female heterogamety. So here females produce two types of gametes. In some gametes there will be autosomes along with the one X chromosome, whereas in other gamete there can be only autosomes and they will lack any sex chromosome, or there will be autosomes along with the Y chromosome. So here we will study two types of female heterogamety, XO type and XY type of sex determination. In order to avoid this X and Y, since we have already discussed X and Y type of sex determination or XO type of sex determination in male heterogamety, here X is denoted as Z and Y is denoted as W. So here in this case the males will have similar kinds of chromosome, whereas in male heterogamety females were having similar kind of chromosomes. Okay. The first type is ZZ and ZW type of sex determination. So here we see female heterogamety. Heterogamety since two different kinds of gametes that will be formed here, whereas males are called as homogamety since they have similar kinds of sex chromosomes, and this we see in most of the birds, fowls, reptiles or fishes. Here we have taken an example of fowl. Here autosomes we can see 76 chromosomes are there, whereas there is one pair of sex chromosome with ZZ chromosome, similar kind in the males. So they will produce similar kinds of gametes having Z's chromosome in the sperms, right? Whereas in females there are 76 number of chromosomes, autosomal chromosomes, and dissimilar kinds of sex chromosome, that is ZW. So definitely they will produce two different kinds of gametes, that is 50% of eggs will have autosomes with Z chromosome and other 50% of eggs will have autosomes with W sex chromosome. So the fusion of these sperms and eggs will determine the sex of an offspring. The egg with Z chromosome when fuses with the sperms will produce males, right? And the egg with W chromosome when it is fused with the sperms will produce female offsprings. Second example of female heterogamety is ZZ and ZO. So here the females will have two types of gametes that will be released, right? So there are autosomes and Z and O type of sex in these individuals. Z and O indicate that there is the presence of only one sex chromosome, okay? It lacks the other sex chromosome. So there are two types of eggs which are formed here, autosomes with Z chromosome and the autosomes without any sex chromosome. So therefore it is denoted as O here, okay? So whereas in male homogamety, wherein as it is said homo, similar kinds of gametes that will be formed since the sex chromosomes are of similar types, okay? So that will be autosomes with the Z chromosome. So here we see that when the males, that is they produce similar kinds of gametes, when they fuse with the Z, that is the gamete or egg which is having Z chromosome, will produce males here, okay? We can see here, right? Whereas if suppose the gamete or egg without the Z chromosome or without any X chromosome, when it fuses with the sperms, will produce always females, okay? So this is ZO here, okay? Some printing mistake. So since females have ZO type of sex determination, definitely the number of chromosomes, the total number of chromosomes will be less in the female compared to males, right? One chromosome will be less in females. That also we should remember here in female heterogamety, that is ZO type of sex determination. There is one more unique type of sex determination which is very important seen in honeybees, and that is called as haplodiploid type of sex determination. That means we will see here haploid type as well as diploid type of individuals, okay, wherein an egg has ability to give rise to males without fertilization, okay, and also it can undergo fertilization and produce females, okay. So we'll see in detail regarding haplodiploidy sex determination in honeybees. So listen carefully. Male have 16 number of chromosomes, okay. Male individual will have 16 number of chromosomes and they are haploid, whereas females will have 32 number of chromosomes and they are diploid. So here males will be always haploid and females will be always diploid, okay. Male individuals, since they are already haploid, they will undergo mitotic division to release the gametes, okay, or sperms. So definitely the gametes or sperms also will be haploid with same number of chromosomes, right? Whereas the females will undergo meiotic division to release the eggs or female gametes. So since it has undergone meiotic division, the number of chromosomes will be reduced to half, right, in the gametes. So each gamete or egg will have 16 chromosomes. So male gamete also has 16 chromosomes, female gametes also have 16 chromosomes. The union of this male and female gamete is called as what? Fertilization or syngamy. Yes. So fusion of this male and female gamete, that is sperms and egg, will give the zygote that will develop into either the female that are sterile. Sterile means they cannot produce any gametes. So those are called as sterile worker bees, or they are sterile females. Okay. So these females will have 32 number of chromosomes. Why? Because there is the union of male and female gamete. So therefore these females will be diploid, right? So the sterile females will be, or worker bees will have here 32 number of chromosomes. Same zygote, okay, union of male and female gamete can also give rise to a queen, which is also diploid. So definitely it will have again 32 number of chromosomes. So here we see that females, both sterile female or worker bees as well as the queen, have 32 chromosomes, right? The peculiarity of this egg have one more unique feature, that it can undergo development, okay, without fertilization, and it will produce the males that are called as drones. So since it undergo the development without any fertilization, and egg itself is haploid here, therefore the males that are produced from this egg will also be haploid. So therefore males will have 16 chromosomes, and the process by which the egg undergo is also called as parthenogenesis. What is parthenogenesis? It is the development of an egg, haploid egg, without fertilization, producing an individual. So that individual here is male. Okay. So we see here that haploid males are formed from unfertilized egg, right? Yes, and diploid females are formed from the union of male and female gamete. So therefore the diploid female or females, they may be queen or the worker bees, will have 32 chromosomes, whereas males which are haploid will have 16 chromosomes. There are two very important points which we should learn here, that is, the male honeybee do not have father. We'll discuss it in detail, but have grandfather. And second statement says males, or they do not have son but can have grandson. So what is the meaning of these two statements? So if we see here that we know that males are haploid, right? So haploid males will undergo mitotic division, and it will give the gametes, haploid gametes, male gamete, and this male gamete will fuse with the female gamete, and it will produce what? Females, okay, which are diploid. Again this female will undergo meiotic division, and it will release the female gametes, and this gamete will develop into the males without fertilization. We should remember that, right? It is called as parthenogenesis. So females give rise to males without fertilization, and males will give females with fertilization, right? So what is the meaning of first statement? The male honeybee do not have father. We'll consider this male. So this male do not have father, right? Because it has formed from female without, that is the gamete that is formed, and this gamete has developed into this male without fertilization. So definitely they won't have father. They can have mother, but have grandfather. The meaning of this is the mother of this male have father, right? Because this female is formed or developed because of the father. So therefore we can say that males do not have father here, okay, but they can have grandfather, right? Okay. The second statement says that males do not have son. So we'll take this male now, okay. This male have daughter, right? They do not have any male son, right? And the second part is, but can have grandson. So this male can have grandson. Because the grandson is developed from the daughter which was produced by this male, right? So this is about Haplodiploidy in honeybees, which is the very important topic, and we should remember these points. We have learned today sex determination, XY type and XO type along with the examples, right? So in XY type, both males and females will have equal number of chromosomes, whereas in XO type, male will have one chromosome less, right, compared to females. Then the number of autosomes and sex chromosomes are equal in XY type, whereas the number of autosomes are equal but sex chromosomes are not, because if we see here that in males one chromosome is less, right? Then in XY type of sex determination, we see that males will produce two different kinds of gametes having X chromosome and another is Y chromosome, wherein Y chromosome is smaller than that of X. Whereas such kind of difference we don't see in XO type of sex determination. Here only one X chromosome is seen in the gamete, whereas the other gamete will have only autosomes without any sex chromosome. But one similarity what is here that we can see that females have similar kinds of sex chromosome, and that is XX. Next was about female heterogamety, ZW type of sex determination and ZO type of sex determination. So here again both male as well as females will have equal number of chromosomes in ZW type of sex determination, whereas in ZO there will be females will have one chromosome less than that of males. Here in ZW number of autosomes as well as sex chromosomes are equal, whereas in ZO type we see that the number of autosomes are equal but sex chromosomes are not. Why? Because here in some individuals we see that they lack one of the chromosome, sex chromosome here, right? So therefore what we see that autosomes are equal number, okay, but the sex chromosomes are not. One unique feature between both these kind of sex determination wherein males have equal number, okay, of chromosomes and they will have similar type of gametes that will be formed since the sex chromosomes are of similar type ZZ, okay. So here in ZW there are two types of gametes that are formed, Z, that is the gamete with the Z chromosome and the gamete with the W chromosome. And in ZO type we see again two types of gametes, but in one gamete there will be Z sex chromosome, whereas in the other gamete it will lack only autosomes that will be present. So that was all about the sex determination. The key points which you should remember here, the difference between male heterogamety and female heterogamety. Then also you should know regarding homogamety too in such kind of species with the examples, okay. And very important topic is with respect to Haplodiploidy or haplodiploid type of sex determination which we have learnt in honeybees, okay. I hope you all must have understood this simple topic. Thank you very much.